It is my pleasure to introduce GeneMedi and Hanbio Group to you today and share some valuable insights. My name is Tristan Kai, and I will be presenting on GeneMedi's viral vector development and non-GMP manufacturing platform. At GeneMedi, we firmly believe that innovation can make gene therapy safer, more efficient, and more affordable. That's what we are dedicated to achieving. After several years of dedicated development, GeneMedi has established unique viral vector platforms, encompassing AAV, lentivirus, adenovirus, covering everything from vector design, cloning, cassette optimization to non-GMP manufacturing. We also extend our services to support scientists in gene editing, including CRISPR vector design, cloning, validation, KO/KI cell line development, and AAV vector-mediated CRISPR in vivo delivery. Founded in 2010 in Shanghai and Suzhou, China, GeneMedi now boasts expertise in vector design, optimization, validation, and non-GMP manufacturing. With 268 employees, over 50% of whom hold master's degrees or higher, we have left our mark in the industry. Our products have been cited in more than 2,000 client publications. We handle over 600 viral vector development projects monthly on our platform, with over 200 industrial and academic partnerships to date. As illustrated in the right graph, our revenue is growing rapidly, particularly from our industry partners. As evident in the following slide, the number of high-quality publications from our clients is rapidly increasing. Our team actively engages in various gene editing projects as collaborative partners. Additionally, GeneMedi is continuously enhancing its capabilities to serve more industry partners effectively. Let's move on to the next part and delve into GeneMedi's AAV vector development and manufacturing platform. Here's a representation of GeneMedi's AAV manufacturing capacity and the stability of different AAV serotypes. As the table indicates, our AAV manufacturing capacity can reach up to 20 liters per batch, with yields exceeding 13 billion Vg per liter in most commonly used AAV serotypes. Almost all widely used serotypes can be produced on GeneMedi's AAV production platform. The graph below showcases the consistent quality performance of our AAV manufacturing platform. Analyzing the yield of our recent 72 batches, you can observe that the yields of common used AAV serotypes, AAV 9, 5, 8, PHP.EB, and AAV 2, consistently fall within a narrow range. The next slide illustrates the high standard of quality control during AAV manufacturing. QC analytics cover titer, purity, safety, and potency. 3 of these QC test items are standard and you order GeneMedi's AAV production service, while others are optional. Notably, qPCR and ELISA serve for titration testing, silver staining for AAV VP1, 2, 3 band helps determine general purity, electron microscope test is an optional analytics for empty/full capsid ratio, and AAV cell-based transduction and Western blot-based expression test are essential for in vitro AAV potency assessment. This slide presents a case of GeneMedi's AAV production and its application in neuroscience. In this case, our client aimed to knock down UROC1 in the mouse hippocampus using AAV. Our team completed all project tasks within 1 month, from AAV-shRNA vector design, cloning, in vitro validation to AAV production. Our client then proceeded with in vivo potency validation, and the results of this work were eventually published in a Cell paper. The next section discusses GeneMedi's capabilities in AAV cassette optimization. The diagram illustrates the elements that can be optimized within the AAV-ITR region. According to the gene therapy vector, elements in red are essential, while elements in blue are optional. At GeneMedi, we can optimize all of these elements. Let's examine a case of AAV cassette optimization involving the WPRE element in the AAV vector. WPRE is a DNA sequence known to enhance gene expression. However, concerns about X protein expression in the C-terminal of WPRE have limited its use in gene therapy applications. Shown here is the full WPRE, the 589 version, which may contain an X protein ORF. Therefore, we designed a shortened WPRE, the 248 version, which is X protein deficient. Subsequently, we conducted validation with GOI's insertion, and we found that the X protein deficient WPRE conferred similar effects compared to the full WPRE. There is another case related to AAV-CTNT promoter vector optimization. The entire project timeline is approximately 2 months and 1 week, covering the selection and cloning of different heart-specific CTNT promoter candidates, AAV production, in vitro assays in cardiomyocytes derived from human ES cells, and in vivo validation in a mouse via intravenous injection. Moving on to the last part of our presentation, the G-NextTM novel AAV development platform. This table presents the tropism in-house test results of different AAV serotypes. Interestingly, we discovered that the AAV8-3M serotype exhibits similar blood-brain barrier crossing and CNS transduction capabilities as AAV-PHP. AAV capsid engineering is the key to developing novel AAVs. There are 2 commonly used strategies in AAV capsid engineering: rational design, which includes specific peptide insertion and structure-based point mutation, and directed evolution, which relies on generating various random AAV libraries, such as point mutation libraries, 7-peptide display random libraries, and DNA shuffling libraries. Each strategy has its drawbacks. Rational design has limitations in candidate diversity and peptide length insertion, while directed evolution, despite its high diversity, often represents low efficiency. On GeneMedi's G-NextTM novel AAV development platform, we employ a combination of AAV peptide display with rational design, which we term rational evolution. Leveraging our knowledge of AAV capsid molecular structure, we can achieve longer peptide insertions. By combining a rational designed peptide library with capsid molecular modification, our G-NextTM platform balances high diversity with high efficiency. Here's an overview of the G-NextTM AAV development strategy. We have built our AAV peptide display libraries using our rational design strategies, calling it the AAV Rational Peptides Display Library. Our libraries are based on GeneMedi's expertise in peptide selection and molecular structure-based capsid modification. For novel AAV assays, we combine screening in unique organic cells derived from human embryonic stem cells or human iPS cells with in vivo screening. Here are 2 cases of novel AAVs developed through G-NextTM. The first is GM400, which exhibits enhanced heart tropism. Compared to AAV9, GM400 reduces infection in the mouse liver and muscles while maintaining similar transduction capabilities in the heart. Another image showcases enhanced cartilage gene transduction capability. All these data illustrate the efficient tissue-specific AAV development on GeneMedi's G-NextTM AAV rational evolution platform. That concludes my presentation. Thank you for watching. If you require this service, please feel free to contact us at any time.